Welcome everybody. Did I get your attention? I'd like to welcome everybody to, I'm not sure which number in the series it is, for the faculty seminars, both Annemieke Verenhorst and Jitendra Pollingwall. We're going to introduce Neil, but they're actually both away and send their regrets. So I'm Rob Curry, I'm the Department of Entomology head currently, and I am kind of teaching in the department, and that's all you're going to hear about me. I'm here to introduce Neil Holliday, who was born and educated in England. You may detect a hint of a British accent still remaining. He got his Bachelor of Science Honours in Zoology at University College University College London in 1971, and PhD from the University of Bristol in 1975, where he worked on population dynamics of the winter moth in apples. He then went to Canada, or came to Canada, I guess, and kind of got an NRC postdoc with Agriculture Canada at the Vineland Research Station, where he stayed from 1975 to 77. He came to the Department of Entomology in 1977 to teach and do research in crop protection entomology, and also became embroiled in a lot of basic ecological and forest entomology research and teaching. He was also very involved in teaching and administration within the agricology program within the Faculty of Agricultural and Food Sciences. He served 3 terms as head of the Department of Entomology and is a hero for helping rejuvenate the department and our department. Supervised 35 graduate students and served on the committees of an additional 67 students. Has a number of research publications, 74 refereed publications and book chapters, and about 350 plus other types of conference presentations, abstracts, and other types of presentations. He received the gold medal for outstanding achievement from the Entomological Society in 2009 and was appointed fellow of the ESC in 2012. He was president of the Association of Faculties of Agriculture in Canada, the Canadian Pest Management Society, the Entomological Society of Manitoba, and the Entomological Society of Canada. He is retired and he is Professor Emeritus in our department, but he still does teaching, research, and service. I'm kind of hesitant to publicly remind him that he's not getting a salary anymore. He's even serving as the secretary. Very valuable member of our department.
I told him to say, and here's Neil. But he obviously felt he had to read what the Dean's Office wanted. And in addition, the Dean's Office wanted me to talk about collaboration, which is why I had to choose the topic I chose. And so for the next, hopefully 50 minutes or less, I will talk about 3 things: an international collaboration that spanned 19 years, some of the biological control research that we did during that collaboration, and we were at a time of change in how biological control was regulated. And so I'm going to intertwine that with the other topics because it had a significant impact upon how things turned out. So that's the plan. So here's my lady beetle example of a predator. This is the parasitoid example. Parasitoid is an organism that has one host during its lifetime, and it develops in intimate contact with its host. And so here we have a parasitic wasp that has an ovipositor right there poking into a host. An egg will be inserted in there, and inside the living host, the larva of the parasitoid will develop until it reaches complete development, at which point it will likely emerge from the still living host, making a hole in it which kills it, and then it will pupate on the outside and produce another adult parasitoid. So, as I said, there's been an evolution in biological control over time, and there's a sort of idea that the 50s were the good old days. And so I thought I'd take an example from the good old days, which also happens to be about my PhD insect, of how biological control was done back then. And in this case, it was done by somebody who many of you will know, Glenn Wiley, who died a few years ago. And he was dealing with the fact that winter moth came to Nova Scotia sometime in the 1930s, it is believed, and by the 1950s was destroying large tracts of deciduous forest, particularly those of red oak. And so Glenn was at Belleville Research Station and was given the great opportunity to do a PhD at Oxford, as long as, in addition, he constructed an inventory of European natural enemies of the winter moth in his spare time. So this took about 6 weeks in the summers of 1952 and 1953, and he was a bit kind of reluctant to do this, but he did it. And as a result, he was able to construct a list of 60 species of natural enemies of the winter moth in Europe. And by the end of 1953, he had come up with a recommended 2 species to introduce. He graduated in 1953. He brought his British wife back with him, and in 1954, in May, he took her off to Bridgewater, Nova Scotia, to introduce the first of the natural enemies for biological control. And it was a great success. It's been repeated many times, and typically what happens is 10 years after an introduction, there's only about 1,000th of the original density of winter moth in a site. But in that 2-year period, there was really very little attention paid to whether there were any risks associated with introducing natural enemies from a different continent. And there are risks, and these are some of them. First of all, we are trying to kill target pests with the natural enemies. But it is quite possible that we will also target or kill non-target pests. And that is certainly something we don't want to do. Natural enemies that are native species may already be attacking the pest we're trying to control. We might disrupt that process by the addition of a new natural enemy. And then we have ecosystem disruption. And to talk about this, I'll deal with the biggest public relations disaster that biological control has ever had. And that is the cane toad, which was introduced into Australia in the 1930s, much at the insistence of the sugarcane producers who put enormous pressure on the local government to do this. So they did. They released about 100 and some odd toads, and they didn't do anything for the cane beetle that they were supposed to be controlling. But like all toads, they have poison glands on the sides of their heads, and they're particularly poisonous in the cane toad. And so they are lethal to the larger predators, the large reptiles and mammals that occur in northeastern Australia, where this thing is spread. In some areas now there are 1 toad. These are 5-pound toads, 2 kilograms and a bit. And the density can be 1 per 5 square meters. So they're easy to eat and they kill lots. And consequently, there's a dearth of predators in these areas, which results in more herbivores and significant changes in the vegetation. So we get a trickle-down effect, and this is ecosystem disruption. So I think biological control is desirable. It doesn't always work, but when it does, it works. But it needs to be regulated. And this conclusion was reached by many of the governments of the first world nations in the late 1990s. And so every one of them put into place some sort of framework for regulation. This is the Canadian framework. I don't want to bother you with the details. It's administered by the Canadian Food Inspection Agency. And it requires that the person who wants to make an introduction produce a petition. And this is considered by a bunch of people who are disinterested in whether this is successful or not, an independent panel, and includes input from the 3 nations in North America. And there are similar reciprocal arrangements by the other nations. And eventually a decision will be made whether to allow the release to occur, not, or we need more data, which we always need, more data. So the petition itself is a very demanding document. The North American Plant Protection Organization has set a series of standards on what must be in a petition. And so this is the document that all biological control practitioners wanting to release something in North America have to adhere to. Everything works towards providing the information this document needs. So I started this collaboration in 1997. The collaboration is with Cabby Europe, Switzerland, Delamont, a mouthful, often referred to as just Cabby, or sometimes the Institute. It had had a long history of working with Canada in biological control activities because many Canadian pests came from Europe. And so a European source of natural enemies to control European pests was logical. Cabby provided the feet on the ground to do things in Europe for Canada, and indeed sent a lot of the natural enemies that ended up being released for winter moth back in the 50s. They didn't do this for nothing. Canada provided substantial funding to Cabby in order that the work be done. During the time I was working with Cabby, we're talking at least $100,000 a year. So in 1997, 2 things came together. One was Uli Coleman was appointed head of the arthropod biological control section in Cabby Delamont. Uli had done a postdoc in California, which might be called the Mecca of biological control in those days. And he was very eager to make a mark in the world for the section and for himself. So that was one thing. And the other thing was bad vibrations about the way Cabby was operating. Cabby was taking Canadian funds and using them to hire summer students and occasionally graduate students from European universities in order to do the work for Canada. And this did not go down well at higher levels in agriculture and agri-food Canada. And so this man, Peter Mason, with Agriculture and Agri-Food Canada, Ottawa, brokered an arrangement by which universities in Canada provided summer and graduate students to do projects paid for by Canada for the achievement of Canadian biological control aims. And so those 2 things came together and we started a program. This is Delamont. This is the Institute. It's a surprisingly small building for the amount of impact it's had on the world. It's 4 stories, full of labs and offices. The attic is now the library, but the most important part of this building, I believe, is the kitchen. This is because the students, graduate and summer students, and many of the staff that are newly arrived here occupy billets in the town. They may have access to the bathroom, but they probably don't have a full kitchen that they're allowed to use. So everybody congregates with a sort of cooperative arrangement to have meals at the Institute. And this happens at lunch and dinner on weekdays and on weekends. So the sort of basic framework under which we were planning to operate was that people would do summer field research in Switzerland, and in the winter they would come to Canada as a graduate student, and they would there do coursework, data analysis, write reports, and eventually write a thesis. Often the original idea was that Cabby would recruit a summer student who seemed to be promising, and the Canadian university would just take this student on. It didn't work out that way most of the time, in fact, because we wanted to have some say in the matter. There was an issue of money. Cabby, for a long time, had been able to figure out how to hire summer students, pay them in Swiss francs, and because they were paid in Swiss francs from Switzerland, they had health insurance through the Swiss plan, and everything was fine and dandy. Well, when we have a summer student, we convert them into a graduate student, we have to find money to transport them across the Atlantic twice a year. We need to find money for health insurance for them. And so the system required that the Canadian person have funds for supporting the student when they weren't at Cabby and for those travel components. And I must admit, I got some rude comments from some of the granting agencies about the size of my travel budgets in order to achieve this while doing research in one continent and having graduate student actually based in the other one. It all worked out. Something that bothered me quite a lot was the idea that a student would have one supervisor for 6 months and another supervisor for 6 months, and this would alternate. It bothered me a lot because I had 2 supervisors when I was a PhD student. Their offices were 50 yards apart, but they didn't always agree, and it was my job to sort of resolve the disagreements that related to my studies. And so I imagine the possibilities when the offices are 5,000 miles apart. And so what Uli and I had to do and agreed to do is we had to meet regularly with the student together. So the student would show up in the spring and get settled, and then I would go to Delamont for a week of intensive meetings where we would meet with the student. I would go to the field sites and get a good understanding of what was going to happen that summer. And then I would go away and things would happen. In the fall, Uli would come to the Entomological Society of Canada meetings in Canada. The student and I would go and we'd have shorter but still intensive meetings to sort out what happened and what the plan should be for the future. And this was critical to the success of this, so that everybody knew what was supposed to happen. It didn't always happen. Life gets in the way, but it did smooth the way a lot. One of the things we didn't expect was to have a different definition of summer in Europe and in Canada. European universities generally start their fall terms around about the first week of October. And so Cabby was used to the idea that summer students worked all September, maybe into October, whereas we were used to the idea that your student would show up for courses in the week after Labor Day. This caused considerable issue, really, and what we had to do was to put all those essential fall term courses into one fall term and arrange for some person in Delamont to take over the research at that point. And for the rest of the time, the student would be a no-show in our departmental seminars in September, mostly. But that's just one of the things we had to figure out. The students had to adapt as well. In Canada, we tend to pay our students by the hour. Cabby paid its summer students by the week. If you can breathe and you can stand, you can work, is Cabby's attitude. Getting up at 7 o'clock in the morning, drive 3 hours to a site which could be in any one of 4 countries, and do research, come back, have dinner at the institute, and since you're there, you can spend 2 or 3 hours after dinner sorting samples. Hopefully not after the Wednesday evening barbecue, which is when everybody got drunk. But the other dinners. And of course, they're there on the weekends. So again, the opportunity for work is always down the corridor from the kitchen. So some of our students were a little less than happy about this arrangement, but they got used to it because there were some compensations. It was only about a 3-hour drive to the Alps. You could go for a hike. Cabby actually organised hikes to the Alps at times. This one was specially for us, but still. And some of the research sites, not unfortunately mine, but some of them were in the Alsace, right next to the vineyards. And so there were opportunities to buy some really nice wines at really good prices. Driving this whole thing is somebody who I characterize as a human dynamo. Uli Kuhlmann was into everything. He was organizing the research. He knew exactly what was going on. He would organize the social events of all sorts. And most people found this okay. Some of them found him a little bit hard to deal with because he was so focused on progress. But I must admit, I enjoyed working with him, and we didn't fight regularly or for any in any negative way. We ended up with positive results. So the example that I'm going to talk about as a product of the collaboration, we actually had 3 projects over the years, is Cabbage Maggot or Delia radicum control in canola, and the possibility of doing biological control in this system. Delia radicum is a European species. It came to Canada and the US probably in the late 19th century, along with some soil and vegetables of somebody who was traveling from Europe to Canada. It had been the subject of a biological control program attempt in vegetables in the 50s. It didn't work out very well. And in vegetables, there are insecticides you can use. I first was alerted to the problem in canola when Rachel Scarth, who some of you may recall was a plant breeder in the plant science department, phoned me up and said, "My breeder's pots are being destroyed by cabbage maggots." And she brought me in some samples that looked rather like this. And plants with no roots don't grow very well, I've discovered. Breeders' plots are only 1 meter or 2 meters wide, and it turns out the cabbage maggot females move in from the side and lay their eggs when they get to the plants. So since the breeder's plot is essentially all edges, they're very vulnerable, much more so than big canola fields. But there is a problem in canola, and in the 90s we didn't know how bad it would get. Actually, we still don't know that. So cabbage maggot at the moment is in these barrel-shaped puparia, inside of which there is a pupa. And in the spring, this will produce a fly, which looks a little bit like a small housefly, and these will feed on nectar for a while, and then the females will move to brassica plants that are bolting and lay their eggs at the base of them. These eggs hatch, and the larvae then move to the root and then tunnel down, browsing initially on root hairs and later gouging the surface and tunneling into the root. This is bad for the root. After they've gone through 3 instars in this environment, they will pupate, usually quite close to the root, sometimes even within it. And if there is time left in the summer, we may get a 2nd or even a 3rd generation, whereas if it is late in the summer, they will overwinter in that pupal stage. So here is the core of the Delia Team, as they liked to be called. This was at an international conference in Davos. This was Hemachandra, who is responsible for actually identifying something we could work on. Lars Andreasen did most of the non-target studies. And Kim Riley also looked at, when we knew what the natural enemy was, how to attract it, and also how it interacted with other natural enemies. And then I have to add a couple of people. Jingdu actually probably never went out of Winnipeg. She never went to Switzerland. She did a lab project for me on how to attract the natural enemy we were working on. And you'll see some of her work. And this guy is Tim Heyer, who is Uli Kuhlmann's right-hand person, and therefore very important, particularly in those September's when the student had to come and do some coursework in Winnipeg. So Hammer's project was to test to see if there were any parasitoids in Europe that were not also in Canada. And he did this by arranging weekly sampling in lots of locations, 6 locations in Prairie, Canada in 2000. And these, of course, were all in summer canola. And in the following 2 years, 12 locations, summer canola, but in Europe, winter canola is much more common, and also in brassica vegetables. And from each of these locations, he would receive a parcel of larvae and pupae that had come from canola or other roots in the field. And he would put them into tubes individually and raise them, hopefully getting either an adult cabbage maggot or a parasitoid out of them. That was success. Failure was when it just died. So success was pretty common. I don't know how many he had in total, but he managed to get success with more than 10,600 individual rearings. Keeping track of each one of them was a major job for him, but he succeeded. And as a result of that, we concluded that there are 3 major common species that we should be interested in. There is one larval pupal parasitoid. So this is a parasitic wasp, Tribliographa rapi, female, that's a female, there's its ovipositor, lays her eggs into a maggot by burrowing down next to the root, getting in there, laying the egg, and then leaving. The larva develops inside the living maggot, and when the maggot pupates, then the larva kills the pupa and pupates within the puparium. There are 2 rove beetles, Staphylinidae. They're very small. Aleocra bilineata, it's all black, and Aleocra bipustulata. And so these are both parasitoids and predators, as you'll see in a minute. So this is a summary, simplified greatly, of that 10,600 successes. And you can see that in North America, Tribliographa rapi and Aleocra bilineata are relatively common parasitoids. These are the percent of puparia parasitized, and there are no bipustulata. In Europe, all 3 occur, and rather encouragingly, since what we might want to do is use that one in summer canola, it does very well in summer canola. So bipustulata looked as though it could be something that we could use for biological control. However, there is a problem. And the problem is that until 1986, there was no species definition for Aleocra bipustulata that was really credible. And it wasn't until 1997 that a diagnostic feature which could be absolutely relied upon was available for saying this is an Aleocra bipustulata. And so we had about 30 publications in North America claiming to have done research on North American Aleocra bipustulata. And so what we had to do was to prove them all wrong. So I wrote to all sorts of museums and tried to track where these people, some of whom have published 50 years ago, would have put their voucher material, which is supposed to be deposited when you do research to allow somebody else to come along and check that you did it on the right species. Hammer went to Jan Klimaszewski's lab in Quebec. Jan is the North American expert on Aleocra, and learned how to use the diagnostic feature. In other words, you have to dissect the naughty bits, the genitalia, in order to determine whether or not it really is an Aleocra bipustulata. Well, I acquired 150 of these so-called voucher specimens, or close to, as close as we could get. And Hammer dissected all of them and sent some off to Jan to make sure he was doing it correctly. And we showed that all of these specimens were actually Aleocra verna. None of them were Aleocra bipostulata, which meant that we could conclude that the distribution of bilineata was transatlantic, the distribution of bipostulata was only in Europe. And therefore, we could introduce it if it was okay to do so. So, something about the Aleocra life cycle. The adults are predatory. My wife would say they're about the size of an eyelash, not very big. And so they eat mostly maggot eggs and larvae. This is 2 Aleocra bipostulata larvae feeding on a 3rd instar cabbage maggot. And when they've had enough food and they've mated, females are desirous of laying eggs, which they do generally close to root maggot puparia. So when the egg hatches, the larva, so little cute and tiny, has about 5 days of life, during which it has the energy to chew through the thick cuticle of the puparium and get inside and start feeding. And if it doesn't do that, it dies. If it does it, then it goes inside, and here you can see the larva inside, and it plugs the hole that it made with a special plug that isn't entirely made up of fecal material, but partly. And there it feeds inside the puparium, but outside the pupa, eating the pupa. And if it's done things right, then it will complete its development and kill the pupa of the maggot, and then it will pupate in the puparium and emerge sooner or later. So we had to determine whether there were other species at risk from this parasitic behavior. And so we needed representative non-target species to test as a sort of panel to evaluate the risk to non-target species in general. And there is a protocol which is very complex that you follow in order to decide what your panel of representative species should be. We started out with a list of 450 species. Totally impossible to work on 450 species. We whittled it down to a list of 18 non-target species, which were flies. We didn't think they would attack anything that weren't flies, which we could get. We needed several hundred of them in order to do the work, and the amount of work involved was considerable. So we ended up with 18, 13 of which were ones that could be encountered by bipostulata in its environment. 5 were chosen because they were beneficial, usually biological control agents that occurred in North America. 7 of the non-targets were quite closely related to Delia radicum. They were in the superfamily Muscoidea. And so, from those 18, we were able to find that only 5 of them, not the bottom one, this is the target species, but the top 5, the non-targets, were actually accepted. That is, the larva went in and was able to complete development and kill the original host in the process. And these 4 of these 5 have very small pupae relative to the puparia of the anthomyiids. These 2 are anthomyiids in the same family, root maggots in other words. And we found that what came out of such a small pupa was a very small bipostulata, which really had very low fitness. And so we could consider these perhaps to be dead-end hosts. We, in addition to the theoretical possibility that if we chuck you in a tube with a potential host, you're going to attack it, we have to know whether the larva would in fact ever encounter the thing that it might attack. So we tried to define the sorts of habitats in Switzerland where Aleocra bipostulata lives. We did this with pitfall traps, some of which had baits, And we were particularly interested in forests because one of those beneficials lives in forests. And so we did this intensively in forests and also in a number of other crops, brassicas, canola, and vegetable brassicas. Allium are leeks and onions, and other veg is other veg. So you can see that they never went into the forest, or at least we never caught them in the forest. They very seldom were encountered in wheat, but they did occur in the vegetable crops. This meant great news for us because one of those species that we had identified as a potential host was Lonkia corticus, and we had another Lonkia species. Lonkia corticus is an important introduced natural enemy for the white pine weevil. And so the fact that it occurs under bark in forests, whereas Aleocra bipostulata doesn't ever go near trees, was very encouraging for us. And so meant that we could at least say, we don't have to worry about this one. The baits that we were using in those pitfall traps were of mustard seed meal. And mustard seed meal was discovered to be an attractant for Aleocra species accidentally by a Swedish guy who was studying organic weed control. And so we decided we'd try to see what role it might play in the biology of Aleocra bipostulata, and could we manipulate Aleocra with this material. So we used a Y-tube olfactometer to study the response of adult Aleocra to test material. So the idea is you pass in air that has passed over a test material, in this case mustard seed meal, on one arm and clean air coming in on the other arm, and then you introduce your insect through the stem and you record which way it goes. And you do this many times. Well, for Aleocra bipostulata, we showed that it was significantly preferred the arm with mustard seed meal that was dry over clean air. It also preferred it over air that had been passing through soil. And it didn't really care whether the mustard seed meal was wet or dry, which is good, because if you use wet mustard seed meal in the field, it's going to get wet. It was important to know. So we wondered whether we could therefore manipulate things with mustard seed meal, and indeed we could. We constructed little plots of canola in Switzerland, and in some, and Linda, my wife, will remind me of the agony of doing this, we scattered mustard seed meal using flour sifters. You know. And so we had a thin layer of mustard seed meal, very precisely added to some plots, and then we had control plots without it. And what you can see here in this graph is that we had a first collection which was at the beginning of the generation of Delia, and a second collection which was a couple of weeks later, and we ran 4 experiments. Always at the first collection we were able to elevate the parasitism level to 30% or more, which was significantly higher than in the control plots. In 2 of the 3 situations where we did second collections, we managed to elevate the parasitism level. So we could use mustard seed meal mulch as a way of increasing parasitism in a particular place, and that would be very useful if we were to release this, because it might release bipostulata. We didn't want flying them in all directions and then not being able to find each other to mate. So it would help us perhaps to retain them at the release site. So we got very excited and we put together a publication which came out in 2007, almost simultaneously with the publication from a French group about an attractant that was a monomolecular odor that was turned out to be dimethyl disulfide, DMDS. So DMDS is produced when brassicas or onions rot. The sulfur compounds in them produce DMDS, and it's also given off when root maggots feed on brassica roots. So we had to find out first whether or not what we had been working on had DMDS in it. And it's convenient to have an analytical chemist as a daughter, because we simply sent some samples to her. It wasn't as simple as all that, but still, she came out with the answer quite quickly that the active ingredient in mustard seed meal is not dimethyl disulfide, because there is no dimethyl disulfide in mustard seed meal. And so we had a project for Jing to do, in which she studied the responses to DMDS of Aleocra bipostulata and also bilineata. We'll just talk about bipostulata here. So she looked at the larvae. Now, the larvae don't live in a windy environment. They live in the soil. So they don't respond in a wide tube olfactometer. So we had to devise a still air arena which has a droplet of DMDS in solvent on one side, a solvent control on the other side, and then we mapped out zones of this many millimeters from that droplet, and we recorded the number of times we observed the larva in these various zones. And this is what you find. The larva spends most of its time in around the DMDS droplets. This is a case of restricted area search. The larva, when it gets into a zone where there is DMDS, walks around in little circles instead of sort of wandering around in larger areas. This we were able to show could influence levels of parasitism by the larvae. We had a different sort of arena. We had provided it with two puparia, one close to a source of DMDS, one close to a source only of the solvent, and 75% of the attacks by the larvae were on the puparium close to the DMDS source. We were able also to do studies on the adults in the olfactometer. And what you can see here, we have females on the left, males on the right, and we have initially 1 to 2 day old newly emerged virgin animals. And these do not like DMDS. They significantly avoid it. Females, after they've mated and are ready to lay their eggs, are attracted to DMDS. Males usually are indifferent, and they were indifferent. They didn't really have any preference. So what does all this mean? Well, I think the avoidance response to the DMDS is because DMDS is produced by rotting plant material for sometimes a couple of years after the plants died. And so it would be very possible for males and females to emerge in a field with lots of DMDS, but no living plants and therefore no Delia. So there has to be a way for them to escape from this field, and so they are at least avoiding, they're not attracted to DMDS at that point. When the females are ready to lay eggs, the attraction to DMDS brings them to a location where root maggots are feeding, mostly, higher probability of that happening, and so puparia, which is the stage that gets parasitized, will be around in a week or 10 days or so, which is about when the eggs of these females will hatch. And the restricted area searching behavior increases the probability that the larvae will stay in the vicinity where there are puparia associated with previous larval feeding. What it doesn't explain is a couple of things. One is, how do the males and the females find each other? First of all, the response to DMDS would suggest they fly off in all directions. And if they fly off in all directions, then there must be something that brings the males and the females together. particularly since these females need to mate about every 10 days in order to remain fertile and produce eggs, which they can do for 70 or 80 days given an available male. The other problem with the idea that they're flying off in all directions and males are indifferent to DMDS completely is that this means they're in other habitats which we have not looked at, and there they may be eating things we haven't thought about. So it's time to think about the adults as predators and what they might be doing there. So we did some studies, again, no choice studies. Lars did these on 29 taxa, which he had selected. And so the no choice means you can choose to eat what we provided you with, or you can starve for 24 hours. So it's a fairly stringent test. These are the ones that showed at least 15% of the tests having predation occurring. And this is a bit dismaying because if you look at all this, we have a mite. It didn't like very many mites, but still. We have some eggs, and we have earthworms, and we have insect larvae. In fact, anything that's soft-bodied seems to be a reasonable prey item as far as an Aleocra bipostulator is concerned. We were happy to note that it always ate the larvae of the target. We were particularly unhappy to note that it also always ate the eggs of a probably beneficial carabid beetle, and very often ate the pupae and sometimes ate the larvae. So this was quite concerning. So Lars continued studying predation in an agonizingly difficult project in which he tried to collect bipostulate adults every 4 hours round the clock in Switzerland and plunge them into acetone, and subsequently using PCR to assess whether or not the guts contain DNA from 2 carabid beetles, we thought, and the cabbage maggot. It took him 3 years to get this working. And the conclusion he reached was that about 20% of the time the gut would contain cabbage maggot DNA that was detectable. And we know that you can only detect cabbage maggot DNA for about 15 hours after a meal before it is sufficiently digested to be undetectable. So this was encouraging, and in fact the first demonstration that in a totally unfettered system these things actually did eat cabbage maggots. But he did some tests on the carabidae and got zeros. He never detected carabid DNA. But neither he nor I have much faith in this, because most of the time carabids are hard-bodied adults that we know don't get eaten because we can put them in the same cage and they don't get eaten. The vulnerable stages are relatively transient, pupae, eggs, and possibly larvae. And it is quite likely with this sort of small sample size that he was able to achieve that we missed those. So there are some big question marks given about non-target predation. Briefly, something on interaction with existing natural enemies. The existing natural enemies in North America being T. rapi and Aleocra bilineata. And for parasitism, it's really very simple. When the puparium is occupied, there is a disincentive and a disinclination for anything else to come in. So T. rapi gets there first because it's a larval pupal parasitoid. Aleocra don't go into those puparia. The plug sends a signal to Aleocra that there is an Aleocra in there. And again, most of the time they don't enter. So the level of interference, if we bring in a new one, a new species, is likely to be very low indeed. For predation, the main target of predation was likely the cabbage maggot eggs, even though we found it convenient to work Usually we're feeding them with larvae because they're bigger. The two predators that are common here already are this Bembidian quadrumaculatum, a beetle, crabid, and bilineator. We know from cage studies that if we add bipostulator, it will add predation, but it will also reduce the efficiency of the existing predators. They interfere with each other to a certain extent. So even though we get more total predation, it's not completely additive. So not too bad a set of news. So to sum up, and I'm going to be sorry about that, we did have a technical hitch, a little later than I had planned at this point. So non-target risks from larval parasitoid attacks, not very likely to be risky for non-anthomyids. A few dead-end hosts perhaps, which probably won't be utilized most of the time. Anthomyids, not so sure, and particularly if those anthomyid flies, the root maggots, are associated with sulfurous host plants, then they're certainly likely to be found. We know about 20 anthomyids well, the biology of them, and they're all pests, and nobody would shed too many tears if Aleocra bipostulator ate them. But there are 600 species of anthomyids in North America, and we don't know anything about the 580 that nobody cares about because they're not pests. Some of them live in remote habitats. For example, there are 162 Delia species. 44 of them are only known from the mountains of northern Yukon. Some people would find it upsetting if they went extinct because we introduced a bipostulator. The non-target risks from adult predation are a real issue. I think these are just optimistically killing anything that they can do and eating it. And because of the response to DMDS, which suggests that they scatter far and wide from where they emerged, it's very difficult to assess impact because we don't know what to be looking for. I think the competition with existing natural enemies is something we can be comfortable about, particularly as these same species occur and coexist in Europe. Something we have not addressed at all is survival in the Canadian prairie climate. But I'm not too concerned about that because they do live in Finland. And I've been to Finland, and it certainly is a vigorous winter. It's quite likely that they would survive here. So, should we petition or should we not? So before answering that question, I need to bring you up to date on the regulatory aspect. Matthew Koch and his colleagues, Matthew was the head of the station at Cabby for much of the time I was working there, produced this study of the level of rate of worldwide introductions of new natural enemy systems from the 1870s to the 2000s, and showed that the effect of starting to have regulation in the 1990s pushed that one down, and this is actually 10 introductions, which is lower than at any time in the 20th century. I'm not saying regulation is bad, but it's a fact of life that we have the same number or possibly fewer biocontrol practitioners working now because it's a little bit out of fashion in some ways because it's so difficult to do, working, and it takes about 12 years now to get the data you need for a petition if you work as a team long and hard. Remember in 1954, 2 years, and you were in the field. So the effect is to make it very difficult and very competitive for what are we going to work on. And so that means that what is getting worked on is the things that are the most pressing, immediate, we got to do something, which at the moment are the spotted winged Drosophila and the brown marmorated stinkbug. It's the major focus of Canadian-oriented biological control out of Cabby, which is where this work would have to be done, because it has to be done in the field where bipostulator currently exists. So I think that what we've done has provided the basis for a future petition, and if the priorities shift, there would be resources to allow these knowledge gaps to be addressed. Priorities to shift would require that Delia become much worse in canola than as it has turned out to be. It may be worse than we think it is, because it's very much a hidden pest, and most producers don't even know it's there. But nevertheless, I think we achieved quite a lot in the project, and it is not lost because we haven't finished the job with the petition. I got out of it this certificate and a bottle of scotch. Some other people got out of it degrees, and there were some publications and some presentations. And another thing I got out of it was a lot of fun. So this work couldn't have been done without an enormous amount of money, which came from a variety of sources, and an enormous amount of cooperation from a lot of people. And it is usual to think that the acknowledgement slide is the end, but you'd be wrong, because this is the end.
Questions?
Sorry it went long, but it took us 5 minutes to fix the text. Yes.
When you get permission to introduce into Canada, do you have to consult the US?
You get permission from one entity to do it in one country, but the other countries have to agree that that it's okay to do it. That's in principle anyway, though in fact in the US some of the states have taken it upon themselves to grant permission for introductions, which doesn't therefore fall under the jurisdiction of the USDA. At the world level, there is no organization. North America has its own, but each other country, well, I think there's a fair degree of harmonization, but there is no single organization that says you can introduce this insect into Indonesia, let's say. Yes.
Do you think that the mustard seed meal is not DMPS? Would it have like something to do with like how we find each other after we disperse?
It's possible. That's in fact a very good idea, because only one species of Aleocra has been studied to determine its sexual communication, and that's a species, Aleocra curtula, which is attracted to carrion. So the first cue is carrion, and when it gets close to carrion, it's such an attractive stuff, you know, rotting mouse, then there are short-range sex pheromones exchanged, and it's a very complex system with multiple pheromone types. So a reasonable model would suppose that plant volatiles would be the long-range cue, and it might be mustard seed meal type things. And then when they get close, there would be some sort of pheromone communication. Mustard seed meal doesn't attract Aleocra bilineata, so it's quite specific to bipostulator. But as I say, I think plant volatiles may be involved in sexual communication at the long range, and then short range would be probably pheromones. Did that answer the question? Yeah. Or did I cut you off before you got to the important bit?
No, that's good.
Okay.
I think everybody's probably in a rush to get out, so we'll cut the questioning off, and we'll take some time to thank you for a really great seminar, Neil. It was really interesting to see the collaboration that developed and the interaction between the various pest predators and alcohol. It was sort of a collaboration from both a student standpoint and a faculty member standpoint. I think it would be a really fascinating thing to do as a graduate student. We're always talking about how we should get our undergraduate and graduate students to have international experience, and this type of sort of interaction between a unit in Europe and Canada, where we trade students back and forth, is awesome, and it's a shame it's not continuing right now. So hopefully that'll be able to be restarted at some point in the future. I do have a mystery gift. I have no idea what's inside here. I was instructed to give it to you, so congratulations. I'm not going to shake your hand.
Oh, yes, of course. It's not